Hello, students. This is Dr. Shazan Mirza. In this video, I'm discussing the use of microarray and bioinformatics and how they both are basically applied in studying the molecular comparison between the species and these molecules, which actually we compare, could either be the amino acid sequences of two different sources or the nucleotide sequences of two different sources. So, and we just try to see, for example, if there are two different species, so how closely both species are linked together, or how close in the timeline did they evolve from a common ancestor and something like that. So let's start with the molecular comparison. So when we are comparing, when we are comparing molecular comparisons of different species of two sources. For example, if you are doing a comparison of amino acid sequences, what do? We use a technique which is called bioinformatics. Now, what is bioinformatics? In bioinformatics, basically we use the protein sequences and we can compare them from two different species or sources already discussed. So, what is bioinformatics? Bioinformatics is an online database. It compares how closely related are the amino acid sequences of two different species or two different sources. So what exactly is bioinformatics? In bioinformatics, basically, we go through the detail that it is a database of gene sequences, genome, protein sequences. When we are discussing protein sequences, it means protein sequences, the amino acid sequences, the primary structures of proteins, and the structure, the 3D structure of the proteins as well. So the biological data is there, and using computer technology and statistics, we try to establish some links and we run some algorithms. This work on algorithms, and it gives us some information where we can apply bioinformatics in biological databases. And how this is applied, there are various databases available online, and these include Uniprot, these include FASTA, Ensemble. Ensemble is one of the major database that we use by putting in the name of a particular gene in the search tab, and with that it gives us the detail of that gene, the alleles and the codons of that. And if you go to Uniprot and you type the name of a protein, it gives you the sequences of the amino acids present there in that particular protein, and what are the variants of that protein present naturally in the organisms. So how these work? Basically, all the database that I have discussed here, or I've pointed these out. You don't need to cram the names of these databases just for information. So how is this applied? So all the bioinformatics information is available worldwide, stored globally. The information is continuously updated, is shared as soon as the information becomes available, no matter which part of world you are in. If you actually you come across a new information in your lab, in your experiments, and that is a valid information and that is valid science, this information gets uploaded in the bioinformatics sources, these databases. It is continuously updated. It runs on algorithms to analyze and interpret biological data. This includes the details of genome, nucleic acid protein synthesis, protein databases. Evolutionary links can be established. This is one of the major ways which we can compare that two organisms, how closely are they linked in an evolutionary timeline. Single nucleotide polymorphism associated with disorders can be studied. What is single nucleotide polymorphism? Single nucleotide polymorphism means a change in a single nucleotide in a codon, which, for example, could be a missense mutation. In missense mutation, what have you studied? We know that in missense mutation, the codon changes. For example, the codon CTG, it changed to CAG. There's a change of one nucleotide T to A, and this is a missense mutation. It can end up coding for a different amino acid. And because of this amino acid being incorporated into the primary structure, the 3D shape of the protein changes. The protein databases store information of amino acid sequences, structures, and functions. How is this applied? What do we do with this? You can determine the structural functional similarity within the same genome or across a species. This can be applied in treatment of disorders. For example, if an organism is suffering from a disease where the protein is not functional or the enzyme is not functional, you can look for an alternate enzyme in another species across different species using this database, and look for an enzyme which actually catalyzes the same substrates. And the gene of that enzyme can then be put inside that host cell to treat that disorder, or the protein from that organism can be given to this particular diseased cell just because two enzymes have a common substrate they work on. So new drug targets can be found. for therapeutics, alternate enzymes, as I've already discussed. For example, enzyme blockers can be found. What is an enzyme blocker? How can they treat diseases? For example, a pathogen causes disease by producing a certain enzyme inside the host cell. Now you know the shape of the enzyme which is there inside the host cell, which is causing the disease. What do? You look for a blocker. You look for a blocker which can block that enzyme's active site. So in that case, the pathogen's enzyme will be blocked, and the pathogen's enzyme will not be catalyzing the substrate into a product, and the product will not be formed, and then the toxicity of the product will not be there, and the disease will subside. So this is one way a new drug target can be found. Apart from this, receptor blockers can be found. What are receptor blockers? We know that pathogens, they penetrate the host cell using receptor. They have proteins complementary to the receptors present on the host cell, so they attach to the complementary receptor. The pathogen enters. All the pathogen, this is they do, they just go and attach to the complementary receptors using the surface proteins. And when they attach to the receptor, they enter the host cell. Once inside the host cell, they cause the disease. Now what can we do? We can look for a receptor blocker. We can look for the structure of the receptor using these databases. We can look for the structures that can come and that can substances that can be used as blockers of those receptors. So if you block that particular receptor, the pathogen will not be able to penetrate into the host cell. A very good example could be the CD4 receptor present on the helper T cells. We know that the CD4 helper T cells have this receptor CD4, and the HIV particle is having GP120, GP41, which is complementary, a glycoprotein which is complementary to CD4 receptor. This allows the HIV to enter into the T cell. Once the HIV is inside the T cell, it starts to multiply and replicate, something that you study in HIV and AIDS. So you probably can look for a new drug target blocking the receptor of CD4 or blocking the GP12041. So in this case, it will not be able to penetrate inside the host cell. So this is how this bioinformatics can be used. Now, going back to what I was discussing of molecular comparisons. In molecular comparisons, apart from the amino acid sequences, what other thing can you study? You can compare the nucleotide sequences as well. Now, when we are comparing the nucleotide sequences, what do we do? The technique that we apply here is microarray. Microarray, what do? You take the cells that are collected from two different sources or species. You extract the DNA from there. You fragment the DNA. The DNA is converted into fragments, which means smaller fragments using restriction enzymes. The nucleotide sequences is then compared using gene chip. How the gene chip works, let's just review this, what we already know about the microarray and gene chip. What is microarray? Microarray is a glass slide, and this glass slide is something like this, that it will have several spots present. This is the microarray glass slide. It's basically not exactly a glass slide, but a slide. So this slide is present. It's just a small piece of gene probes attached here. So this you can see is a gene chip or a microarray. So I am just drawing this same thing here at the top to show how this gene chip actually works. So the microarray is having several spots. Every spot is a gene probe. So this is a separate gene probe. The one below is a separate gene probe, and the one below it is a separate gene probe. So you have as good as 1, 2, 3, 4, 5, 1, 2, 3, 4, 5, as good as 25 different gene probes present on this microarray. What exactly is a gene probe? Gene probe is a single-stranded sequence of bases to which a single-stranded DNA molecule can come and attach. So how can that happen? I use the term DNA molecule, though, although that can't apply. So what exactly is a gene probe? Gene probe is simply a single-stranded sequence of bases. So for example, this is a gene probe, single-stranded sequence of bases. This base sequence will be complementary to a sequence present on the allele or the gene of the template strand of the DNA molecule. So what do? We take the DNA molecules from two sources. For example, we fragment the DNA, we cut them. When we cut them, we convert the DNA molecules into single-stranded structures. We need single strands. So the single strand having a complementary sequence will come and attach at the respective gene probe. We can label the source of DNA with two different color tags. For example, red and green, we can tag one as red, taken from one source. We can take the other source and tag it with green. And then we can pour these single-stranded fragments onto the gene probes. Once you add the sample, for example, I'm adding the red sample first. So every gene probe will allow the respective complementary sequence to attach if it's there on the gene. So it will come and attach when the single-stranded segment of the DNA attaches to the gene probe. We say that the process of hybridization is occurring. What is hybridization? Hybridization is the attachment of a sequence of gene probe to its complementary sequence on the DNA single strand. For example, this is hybridization. This is the region where hybridization occurred between the gene probe and the green tag DNA. Here you can see there were two gene probes. This is one gene probe. This is another gene probe. Both gene probes have the same sequence and they are present on the same spot. So for example, this is the spot. In this spot, I had two gene probes of the same sequence. One of them allowed the green colored DNA strand to be attached to be hybridized. On the other, the red one got hybridized. So this one gene probe will end up giving me a color combination of both, showing that this particular gene is present in both the sources. So what do we do? How do we go about it? We allow the DNA samples to be added to this gene probe, to the microarray. Once they have been added, we allow, for example, the once I added my DNA, the red DNA, then I washed it, then I added the green DNA, then I wash it again. And once the DNA were hybridized, I will wash off the unhybridized fragments. Why so? So that any fragment that was not complementary to the gene probe, it will be removed from there. So this is how a gene probe, a microarray slide can look like. Wherever we see a yellow spot, the yellow spot actually shows that this gene is common to both species. The red colored DNA and the green colored DNA, they both hybridize on these spots. A green spot means that only the green tag DNA hybridized here, the red tag did not hybridize. And at the red spots, we know that the red tag, the DNA which was tagged red, it hybridized and the green did not hybridize here. And the spots that appear blue are the gene probes that did not have the complementary sequence attached to them, which means that every blue probe here represents a gene that is absent in both the sources. At times, we see an orange spot, or you can see a spot which is not exactly yellow, which is more towards green. So this orange spot, what does this say? So for this, we need to just recall a particular fact regarding microarray. That microarray on microarray, there are defined position of tiny spots with each spot containing a known DNA sequence and how many copies of that DNA sequence are there, multiple copies of the same DNA probe. So for example, if this was the gene probe, this was although one spot, and this spot is having a single gene probe, but it will have many copies of the same gene probe being present here. So it can allow both colored DNA tag to be attached here, to be hybridized here, but it can also quantify which actually got hybridized more than the other. For example, here we are seeing a color in a spot which is orange. It shows that on this particular gene probe, the red tag DNA got hybridized more than the green tag DNA. So this is how the microarrays work. Apart from the molecular comparisons that we have just discussed, so what else do we need to I'll need to discuss here? I'll be discussing the detail of mitochondrial DNA as well here. So let's move on to that. So when we are discussing this, for example, microarray, before I go to mitochondrial DNA, just recall microarray once again. So in microarray, we are doing molecular comparisons. We can discuss evolutionary studies as well. So we are Homo sapiens. If we compare the DNA sequence of ours to the ones which are regarded as Homo neanderthals, what are Homo neanderthals? Homo neanderthal, we are Homo sapiens. Homo neanderthal and Homo sapiens, they both come from the same genus, but the species is different. They were Neanderthals, we are sapiens. You can look for a very good documentary, Secrets of Neanderthals. It's just 1 hour, 20 minutes available on Netflix. It's a very good introduction to Homo neanderthals if you have any interest. And I do recommend this to my student that they should go and just look for the details of how Neanderthals are about. So anyhow, coming back, so what you have, what how can we use microarray here? So you can take the sample of cells from you can actually extract the DNA from the remains of Homo neanderthals, and you can extract the DNA from the Homo sapiens as well. You have two sources of DNA now. You can fragment their DNA. You can tag one of them as red. You can tag the other one of them as green, and you can add them on a microarray to find out how many common alleles or genes do we have. So we have a very similar DNA. The DNA is so similar that it appears that our evolution, actually the split occurred sometime around 500,000 years ago. They have not the amount of variation that have accumulated over the years. This accounts for about this much time has passed. So since this split occurred, Homo neanderthals are extinct now. If we compare our DNA to the living organisms here, so 98% yellow spots can be seen if we compare our DNA with chimpanzees. So coming back, let's move on to mitochondrial DNA. What exactly is mitochondrial DNA? Mitochondrial DNA is present inside the mitochondria. It follows a maternal lineage. So even mitochondrial DNA can be used as a source of finding out the ancestors or common ancestors or evolutionary links. And why mitochondrial DNA can be preferred over the nuclear DNA? We know that cell DNA comes in nucleus and mitochondria both. In nucleus, we have DNA that is having two copies of every gene, but mitochondria will have several copies of the genes present inside the cell. So a mitochondrial DNA, because every mitochondria will have at least one of the circular DNA, and the more the mitochondria are there, the more the circular DNA will be present here. All will be having the same genes. So extracting those genes is very easy. And there are smaller number of genes present of mitochondria, which can easily be studied. The mitochondrial DNA comes from the maternal lineage only, so this can be traced back to mothers and the mothers of the mothers and so on and so forth. So this has been traced back, for instance, to a single woman, the mitochondrial Eve, who used to live about 150,000, 200,000 years back in Africa. And this has been done because the mitochondrial DNA is studied that the mitochondrial DNA mutates at a very regular rate. It mutates about once every 25,000 years. Unlike the nuclear DNA mutates randomly because there are a lot of random mutations that go on. So you can't really study a lot of variation accumulates in the nuclear DNA, and the variation that have accumulated in the mitochondrial DNA is very small, and it can be used as a source to study how the evolutionary links work. So there is no recombination occurring. It's coming from maternal lineage. There are many copies of the DNA available in the mitochondria. It mutates at a very regular rate, and it is circular DNA. There are no histones present there. So that's all I wanted to cover in this video. I hope this will be helpful for you. Thank you so much.